A G protein linked receptor is a plasma membrane receptor that works with the help of a G protein. The G protein is loosely attached to the cytoplasmic side of the membrane and acts like a switch depending on whether GDP or GTP is attached to it. When GDP is bound to the G protein, it is inactive. When a signal protein binds to the G protein linked receptor, the receptor changes shape, causing GTP to displace GDP. This activates the G protein. After the G protein is activated, it disassociates from its receptor and binds to the inactive enzyme. The enzyme is now activated. The enzyme now catalyzes the conversion from ATP to CAMP. The CAMP is now a second messenger activating the protein kinase. The protein kinase goes on to activate the signal transduction pathway. The cellular response is the release of calcium ions from ER into the cytoplasm. Tyrosine kinase receptors are plasma membrane receptors that attach phosphates to tyrosine and once activated produce transduction pathways and cellular responses. Tyrosine kinases are individual polypeptides with ligand receptors outside of the cell in an intracellular tail containing multiple tyrosines. When signal proteins bind to receptors, the two polypeptides associate closely with each other, forming a dimer. This activates the tyrosine kinase region of each polypeptide. Each tyrosine kinase adds a phosphate from ATP to a tyrosine on the tail of the other polypeptides. This is known as the phosphorylation cascade. The tyrosine kinase receptor protein is now fully activated. Because of this, inactive relay proteins bind to the phosphorylated tyrosine, thus undergoing structural change to become activated relay proteins. This triggers a signal transduction pathway, leading to a cellular response.